It's 9:30 and it's time for another ELIXIR Coffee. And today it will be Josef Pánek from Institute of Microbiology from the Academy of Sciences, who will be speaking about the template-based prediction of RNA secondary structure using his copy-paste algorithm cpPredict. Pepo, the floor is yours.
Okay, so good morning, everybody. I'd like to add to the introduction that, the main purpose of template-based prediction for RNA secondary structure is to use existing experimentally identified structures of RNA molecules to predict secondary structures of RNA molecules without known structure. It's because, to employ the existing structures, it's because, to identify secondary structure, or in general structures of RNA molecules, is extremely difficult because RNAs are unstable and it's difficult to extract enough RNA for its structure to be experimentally identified. RNA molecules are extremely unstable and they are degraded fast by enzymes, RNases, that are everywhere ubiquitous. That's why it's so difficult to get sex structures of RNAs. I believe 2 or 3 years ago in PDB, I don't know how it is now, but there were not more than 50 or even 40 only structures of RNAs, and most of them, or maybe even all, were identified in complex with proteins that stabilize the structures of RNAs. So that's why such methods have their use. And yes, the idea of cpPredict, which is a nickname for copy and paste prediction, is it starts with multiple sequence alignment. In this multiple sequence alignment, there are 34 mammalian RNAs. In this case, they are 18S ribosomal RNAs that are pretty long. They have almost 2,000 nucleotides. And in the lines of the alignment, which is this, there are sequences of RNAs, and in columns there are nucleotides, aligned nucleotides, and that are colored according to their type. And this stripe below, it shows conservation of nucleotides in individual colors. In the alignment, you can distinguish consistent blocks, for example, as this one, that one, or this one, and that one again, in which sequences or subsequences, sequence segments of the whole, like sequences, are very similar. And the idea is, if they are similar, then also the structures, the structure that they code for should be similar. So if you have one of these RNAs aligned in the alignment have a known structure, as in this case, it's a human 18S ribosomal RNA. So you can use the structure segments coded with these similar sequences in consistent blocks for copying it onto sequences of other homologous RNAs. Because if they are homologous, it means they have the same function in different organisms, and therefore they should have similar structures, also secondary structures. And if these sequences are similar, the structure segment that they code for should be also similar in RNA molecules of homologous species, or evolutionary closely related species, as in this case. So we simply take, or relatively simply take the structure segments that should be similar in other molecules and copy it onto sequences of other RNA molecules according to their positions in the alignment. In this case, there are a few of such similar structure, evolutionarily conserved segments that are colored. And yes, and for the inconsistent blocks that contain dissimilar sequences that code for dissimilar structure segments, as in this case is this segment, this one, and that one, that have different structures, you know, in the similar species because of their function or something which can be specific for a species. So their structure of these like structure segments is predicted de novo using individual sequences for every RNA and only the sequences that are in the inconsistent blocks. And these newly predicted structures are pasted to the like copied structural blocks, and they are matched together to form the resulting predicted structure. You know, this worked quite well, but you know there was a big problem that we could not like sort out well enough, and it was like identification of boundaries between consistent and inconsistent blocks. And if you are not able to identify exact sequences of these consistent blocks, then you copy like a structure segment onto a wrong sequence, and the result is a nonsense. So that's why we used pairwise sequence alignment. This is another example of RNA. This is a template structure, which is SAM1. It's also taken from TDB, and we try like using this template predict a structure of homologous RNA, which is the same RNA with the same function, but for different evolutionarily related organisms. And after the copy step, as I showed you before, but only using like two sequences aligned, the alignment, yes, the alignment is in detail shown here. This is a sequence of template RNA, which is aligned to a sequence of query RNA, and the alignment is shown with these like vertical lines, and the secondary structure of the template is shown here with these base pairs. And these base pairs are copied according to the aligned nucleotides onto query sequence. But you know, there is a couple of situations in which base pairs cannot be copied. You know, for example, this base pair CG cannot be copied on the query sequence because there is a G is missing here, and there is a gap in the aligned sequence. So you have to like work out all these situations, but mostly these base pairs can be copied like meaningfully on the query sequence because the RNAs are homologous. It means they have similar structures. And the result of this copy step is something that we call an intermediate structure, in which some parts are well formed, for example, this hairpin, and others are badly formed, like for example this one. There's a lot of gaps in this structure, and AG and nonsense like base pairs for secondary structures, or even this stem. You know, you can see that CC is paired with CC, and there's a gap in another one. So you have to treat this intermediate structure to get a correct, like biologically valid structure. Yes, and it's done using decomposition of this intermediate structure to its like basic structure elements, and that are in this case 3 hairpins, this one, that one, and this one again, and one stem like structure. And for these elements, their consistency, structural consistency is computed. And for inconsistent elements that are in this case this one and that one, their structure is predicted de novo using their sequences, using like existing tools like RNA fold and RNA duplex. We use RNA duplex for stem-like structures and RNA fold for like hairpin-like structures. And then these newly predicted elements are matched to together with consistent elements, and they form the resulting predicted structure, which is in this case, you can see this is pretty similar. They are homologous molecules, and yeah, the only difference is in the length of the stem and in this hairpin. So this is the principle of the prediction. So finally, we used for cpPredict, we use pairwise sequence alignment because it's like it doesn't have the need for like for identification of the boundaries between consistency and inconsistency in the line sequences. Yes, so this is we tested cpPredict efficiency with by comparison to other algorithms for prediction of secondary RNA secondary structures that were in this case refold method, RNA fold, and R-search-based method. All these methods have different amount of information as its inputs. For example, RNA fold uses as its input only a query sequence, whereas, for example, the refold method, as well as cpPredict, they use much higher amount of information, which is like a template structure, template sequence, and template query sequence. So they are supposed to perform better, you know, like in prediction. And they are compared in these two schemes. And we tested them like with prediction experimentally identified structures, and we compared the predicted structures to their experimentally identified counterparts. The structures were taken from PDB. In this case, we compared whole structures predicted and experimentally identified using edit-tree distances. So it means the less number, the higher accuracy. And in this case, we compared a percentage of correctly predicted base pairs. So it means it's the other way around. The higher number, the higher accuracy, and both these diagrams show that cpPredict performed best in this test. RNA fold predicted worse, but you know it's expected because you know it uses only a query sequence as its input. But anyway, cpPredict, you know, performed better, even better than refold method or RNA search method that uses the same amount of input information. That's what we wanted. Yeah, we made a web server for this algorithm. This is its input page that you put a template sequence and template structure in a dot bracket form and query sequence, and then you can like set up few parameters. I will come to them later. And this is an example of output page that shows prediction using the same template, which is shown in this like left side windows, which is in this case bacterial RNA, which is success RNA from Bacillus subtilis, and the predicted structures are shown in right side windows. And this is an example of prediction that the upper for homologous molecules and non-homologous molecules. The upper part showing non-homologous molecules, where using this template, the structure of RNA, which comes from evolutionarily distant organisms, have different function, different structure, and also dissimilar sequence is predicted. And as you can see, the predicted structure is really bad. But it doesn't mean that the prediction is wrong. You know, it's not, because simply, you know, this sequence cannot adopt this structure. And in this case, it's clear. But you know, there are cases, there are predictions that it's not that clear if the structure is well formed, the predicted structure is well formed, or it's bad. You know, so that's why we test predicted structure, or we like put in as a part of cpPredict possibility to test predicted structures using Bootstrap. When we randomize 100 or 10 times the query sequence, predict their secondary structures using exactly the same way, same template as for regular query structure, and then we compare both predicted and regular predicted structure and randomized predicted structures to the template and compare their similarity. If the similarities are similar, it means that there is something wrong, and most likely that the predicted structure is not biologically valid, and the comparison produces z-score, and in this case, the z-score is negative, which tells you that the structure most likely is not biologically valid. This is just an example, extreme example. And of course, because it's the sequence of this query RNA comes from plant, which is evolutionary far from bacteria, Bacillus subtilis, and this RNA does not exist in plants. And this testing, besides cpPredict can do this template-based prediction, it also can be used for testing if the query sequences can adopt, a particular structure, which is quite useful if you have sequences of unknown RNAs and you want to know if they are homologous to the template RNA. So you try to predict their structures, and if the sequences cannot adopt the structure, secondary structure of template RNA, it indicates that they most likely are not homologous. It means that they have different function, can come from different organisms, and so on. Whereas if you predict secondary structure for RNA which is homologous to template RNA, the prediction is very nice. And in this case, there are two bacilluses. It means they are the RNAs comes from very closely related organisms, and the prediction is nice. And it's also, again, it's indicated this z-score, which is really highly positive. Okay. This is last slide. So this work was done in collaboration with David Uza from Charles University. Here we published the first version of cpPredict based on multiple sequence alignment. This is pairwise sequence. The version based on pairwise sequence alignment. This is the paper on web server. And finally, we made a database that contains a predictive database that contains structures, RNA structures, secondary structures predicted using cpPredict and using templates taken from PDB. I don't know how many RNAs are there, but the advantage of these RNA is these RNA structures is that they are predicted for individual RNA structure. For example, in RFAN, you have also predicted secondary structures, but they are consensus secondary structures. And these consensus structures, even if they gave you, or even if they give you some information about how secondary structures looks like, for individual families, it's still some consensus secondary structure that can be different from individual structures, or from structures of individual RNAs that are included in the consistent structure. And yes, the structures are viewed using Traveler, which is based on the same in the same principle as the CP prediction, and that you could hear about last week, I believe. So that's it. It was fast, but that's it. Now, if you have some questions. No, I can't hear you.
What templates can be used? Only the templates with the structure, or basically any multiple sequence alignment?
No, it's only single structure.
Mm-hmm.
Secondary structure, only one. The alignment is made between this template sequence, between the sequence of the template RNA and the query RNA, and then the structure of the template is copied and pasted onto query sequence. I don't hear you again.
RNA of the SARS-CoV-2 and the other coronaviruses somehow?
Sorry, could you repeat it? I couldn't hear you, you know.
So is it possible to visualize the RNA of the SARS-CoV-2, or at least of some parts of it?
Oh yeah. I don't know if they are included in the rPredictor DB database. If they are not, you know, because it's the database created by Josef Pánek from our lab, you know, it's a David Hosa students. I have to ask him first if he already put these RNAs in the database.
Okay. So, any other questions from anyone? I don't know if you've already replied to Marian, his questions about the flexibility of the RNA structures.
I see it. How flexible are these RNA structures? Can you assess what parts are flexible? No, you can't. You can't using these algorithms. Yeah, it's a good question. It would be nice, you know, like to have this possibility, but you know, no, you can't. Using these algorithms and this predicted structures, you know, you can't do it. No.
Okay. Any other question? If not, then thank you again for the nice presentation and showing us how the program works and what we can learn from it. And next Friday, it will be Jiří Hon from ICRC, who will speak about the enzyme miner and SoluProt mining for soluble enzymes. And with that, that's all, and see you next week. Bye.